Afternoon. Thank you for participating and coming to the GI Grand Rounds. And it's my pleasure to give you some thoughts about the leaky gut, reality, folklore, or is this a new frontier? So what I'm planning to do is to give you some understanding of the components of the intestinal barrier, review how we measure intestinal permeability in vivo in particular. And then I'll go through some of the literature on the effects of stress and diet on intestinal permeability, the effects of symbiotics, prebiotics, and probiotics. Very briefly review some of the literature on diseases or disorders associated with intestinal permeability, and inevitably, those same reports also document the effects of altered microbiota. And then I'm going to finish off with some futuristic looks that I think are really quite exciting with regard to barrier dysfunction in the context of inflammatory bowel disease and potentially new molecules that work on the leak pathway. So for those of you who might want to read more about this, I've just put up 3 of the papers we recently published, and you see the PMID numbers. And if anybody wants copies of these PDFs, please send me an email and I'll be happy to share with you. So let's just go back and think about the components of the intestinal barrier. This is a nice review article by Kata and Soderholm, and it shows that we have in the lumen certain factors that help with the degradation of bacteria and antigens, such as bile, gastric acid, and pancreatic juices. Then we have the commensal bacteria that inhibit the colonization by pathogens. We have a microclimate close to the intestinal epithelial layer and the microvillus. This consists of the unstirred water layer, the glycocalyx, and mucus. The mucin layer, as we'll see on the next slide, is extremely important as a protector as part of the barrier. Then we have the epithelial cells themselves, and they are associated with junctional complexes, and they also serve to keep injurious agents out. We will go through the types of junctional complexes, but also we'll think about the paracellular, the defense mechanisms between the cells that are sometimes lost, and therefore increased permeability occurs. Let's not forget the innate immunity, the acquired immunity with immunocompetent cells in the lamina propria, and of course the enteroendocrine cells and the enteric nervous system, which stimulate motor function and propel the content which may contain dietary or pathogenic antigens down the GI tract and out through the stool. Let's spend a little bit of time thinking about these defense factors. I think we have to remember the importance of the mucin layers. There's a firmly adherent inner layer of mucin and then a thicker but more loosely adherent outer layer of mucus. And these have a pretty significant thickness such that there's quite a distance for something that's in the lumen to get at those epithelial cell layers. Then there's the unstirred water layer, which I described to you in the previous slide. We have to keep in mind that there are innate variations in permeability. The small intestine is far more permeable than the colon epithelium. And of course, for those who might want to know some trivia about permeability, the most permeable gastrointestinal organ is actually the gallbladder, which, as you know, concentrates bile with a lot of water being reabsorbed through the gallbladder epithelium. The epithelial cell layer itself has transmembrane mucins and the glycocalyx. And then there are the immunocompetent cells, including the Paneth cells, producing lysozyme and antibacterial peptides. And as we said before, the secretory IgA produced by the immunocompetent cells in the lamina propria. I like this very simple cartoon that comes from a review by Stefan Bischoff. And it shows these cells that we need to think of in the epithelial layer. The enterocytes, the Paneth cells, which, as we said, produce defensins and lysozyme, for example. Then the goblet cells, which synthesize and excrete mucus. And then the important dendritic cells, which are sampling for the antigens in the lumen. And then, of course, the immunocompetent cells, the intraepithelial lymphocytes, the plasma cells, which are also serving as defense mechanisms against injury. And then this final little mental image that I find useful, which is that there are pores which allow smaller molecules like ions, water, and smaller molecules, which we'll describe in the next few slides as part of our probe molecules for testing permeability. Larger molecules like antigens and macromolecules goes through apoptotic leaks or erosions and ulcers. And of course, let's also remember that there is transcellular permeability, if you wish, or transcytosis, whereby larger molecules are, including amino acids and peptides, in fact, are able to be taken up by specific transport processes, go through the molecule, and then pass through exocytosis down into the lamina propria. So how do we measure leaky gut? There are several potential approaches, but the two that I'm going to talk about mostly this afternoon pertain to the urine excretion of probe molecules that are taken orally. And these are very suitable for in vivo studies in whole humans, which is my area of interest. And then the appearance in serum of bacterial lipopolysaccharide, or indeed the binding protein for the lipopolysaccharide. So there's an assay in serum for LBP, it's called lipopolysaccharide binding protein. And then there are substances that are endogenous to the intestine, like zonulin or intestinal fatty acid binding protein, which on the occurrence of epithelial damage will find their way into the peripheral circulation. So these are the main approaches that have been used to measure leaky gut in vivo. Now, again, this is an exhaustive list of the potential types of methodologies to measure intestinal permeability. And as I said, urinary excretion of probe molecules assesses the function of the entire barrier. And then the serum, bacterial lipopolysaccharide, zonulin, intestinal fatty acid binding protein, also they tend to reflect the entire barrier function, and particularly the potential for damage to the intestinal epithelium. There are, of course, many other approaches to doing this type of study, and this involve either organoids, mucosa taken from the human intestine and studied in vitro, and also morphological studies or expression studies. And there are also other in vivo studies that can be done, like mucosal impedance endoscopically or laser confocal endomicroscopy, which I won't be describing today. They don't really lend themselves to large population studies, which are more relevant, in my opinion, in the context of dietary and other interventions that are being proposed for the leaky gut. You have to keep in mind that the probe molecules have different molecular sizes. This is Ångström himself. And of course, the molecular diameter of these molecules is measured in angstroms. And there are in the literature examples where the actual diameter of these molecules have been reported, or they can be estimated based on a fairly standard approach to estimating the molecular diameter of a molecule based on its molecular mass. I want to draw your attention to 2 or 3 sugars in particular, because they are the ones that we're going to be discussing. Rhamnose is a monosaccharide with a diameter of approximately 6.9 angstrom and a molecular weight of 164 Daltons. Mannitol, monosaccharide, 182, 7.2. Lactulose is a disaccharide. You can see here the doubling of the molecular size. But notice that the diameter increases relatively small amount, but this is quite relevant, as you will see soon. And sucralose, which is another sugar disaccharide that is being used, approximately the same molecular mass as cellobiose and lactulose, and a slightly enlarged diameter. Now, keep in mind that when you look at the literature, some of the things that are being measured, like bacterial endotoxins or lipopolysaccharides, they have a much larger estimated diameter based on their molecular mass. And also, there are experiments that are done mostly in vitro studies in Using chambers with things like dextran 4 kilodaltons or 40 kilodaltons and even 70 kilodaltons, that obviously are looking at a very different molecular size. Notice even the 4,000 dextran, which is often linked to FTSE, and that has a molecular diameter of about 30. Now, why is this relevant? The molecular size determines how much actually comes across the relatively healthy epithelium. Now, obviously, if you have celiac disease or Crohn's disease with ulcers, you're going to have a much greater propensity for larger molecules to go through. But these experiments, which were done by Ingvar Bjarnason many, many years ago, illustrate, I think, very nicely the proportion, the percentage of these sugar molecules or other molecules, chromium 51, EDTA, and polyethylene glycol, how much actually gets across if you give these molecules by mouth and you collect the urine over the next several hours. So as you see, there is almost like an inverse proportion with the diameter and also with the molecular size of these probe molecules. And we need to keep this in mind. I'm going to come back to this point again later, but you'll see that lactulose, there's probably less than 1% of the lactulose that's actually absorbed and excreted when you give it as a permeability probe molecule. So there may be confounders with these oral probes. First of all, is there bacterial degradation of the sugars? Of course there is, and the one that's least degraded is sucralose. And it has been proposed that sucralose may be a very good molecule for colonic permeability. But we also keep in mind that sucralose could be a molecule to assess small bowel permeability. What is the best time for collection of the urine to reflect small bowel and colonic permeability? Well, studies we did here at Mayo by using radioisotopic labeling of the same drink of water that we gave showed that by taking images of the abdomen while we were doing the urine collections, for small bowel, the urine collection that reflects the small bowel is between 0 and 2 hours, and for the colon between 8 and 24 hours. And the collection that we did between 2 and 8 hours generally reflects both the small bowel and the colon. Does the dose of the probe molecule impact the result? Potentially. Well, the answer to that is if you give lactulose or indeed even mannitol 5 grams or lactulose 10 grams, you're likely to induce secretion, osmotic secretion in particular, that then can give you a much more rapid transit through the bowel, and you don't know really whether you're studying small bowel or colon. And so we worked hard to develop an HPLC tandem mass spectrometry measurement here at Mayo Clinic with the help of our colleagues in the immunochemistry core lab, Ravinder Singh and colleagues, in order to reduce the amount of lactulose down to 1 gram, which is not going to perturb the intestinal fluid and transit profile. Is there environmental contamination? I'm going to show you data that shows that this is a very, very significant confounder, particularly for mannitol, but also for some of the other sugars. And then keep in mind the potential limitations of saccharide ratios. Most of the literature, you're going to find lactulose to mannitol ratio, lactulose to rhamnose, sucralose to lactulose. And I want to keep your attention to the fact that if you don't have a grossly inflamed or ulcerated intestinal mucosa, you can end up with only about 0.5% of the mass of the disaccharide actually absorbed. And I'll show you those data in the next few minutes. So we just had this paper published by Gastroenterology in which we did some validation studies in order to try to develop a test that is robust enough to propose not only for research studies, but also simple enough also to implement it as a clinical a type of study. So we actually did studies in which we administered monosaccharides, 12 carbon mannitol, 13 carbon mannitol, rhamnose, and the disaccharides, sucralose and lactulose. And then we did studies in 60 people aged 18 to 70, both males and females. And we did studies containing 16 grams of fiber on 2 occasions and 32 grams of fiber on the 3rd occasion. So let me try to summarize what we found. The first important point is that mannitol is a very significant confounder. So even before, this is the baseline urine that we take before we gave 500 milligrams of mannitol, you'll see that there already was mannitol in the urine. And in fact, there was in some people up to 50 or 60 milligrams of mannitol in the urine before we gave this as the probe molecule. Now below, you'll see that we've done studies with 16 grams of fiber twice and 32 grams of fiber once, because we wanted to see, does a variation in the amount of fiber taken in the diet actually impact the results of the permeability measurement. Now, this is the amount of mannitol we gave, 500 milligrams. Based on the 13 carbon mannitol excretion over 24 hours, this is the amount of 12 carbon mannitol that we would have expected on average. And what you can see is that over 24 hours on all three tests, 75% of the people had more 12 carbon mannitol than we would have anticipated based on the similar molecular mass of 13 carbon mannitol administered. So in other words, all of this is really contamination from the diet. Now, keep in mind, these were studies done in the clinical research trials unit where we controlled the diet over the period of 24 hours before and the 24 hours when we were doing the collection of urine. And so this is a very significant contaminant. And I bring this to your attention because there's so much of the literature which does not actually correct for any potential contamination, even in these very carefully done balanced studies that we did. These are some data of the 4 sugars that we studied. And as you can see, here we have 13 carbon mannitol. Here we have rhamnose, lactulose, and sucralose. I'll just make the point again that with the disaccharide lactulose, we gave 1,000 milligrams, and the total amount on average was about 5 milligrams. So it's 0.5% of the mannitol, of the lactulose that we gave by mouth that actually was collected in the urine. That's the median amount, okay? There are some people with a little bit more. And similarly, remember, sucralose would be great as a marker for, particularly for colonic permeability, but again, the amount of the disaccharide that's absorbed is only about in a few percentage points. Our studies suggested that the optimal probes were 13 carbon mannitol and lactulose. Why do I say that? I say that because we also found contamination at baseline with rhamnose and of course with sucralose, which is used as something to sweeten so many foods and so many drinks that it's very difficult to exclude it if you're doing these types of studies. So on balance, we felt that 13 carbon mannitol and lactulose, particularly using lactulose at only 1 gram total amount, would be the optimal probes for measuring permeability. Because we studied an equal number of males and females, we were able to check whether there were any differences by sex, and there weren't. And we also had three groups by age, and you see the age ranges here that we studied, and there was no difference by age up to the age of 70. So we feel we have a good basis. We have normal values, and we have also characterized the fact that there is no effect of doubling the amount of fiber in the diet. There's no effect of sex, and there does not appear to be an effect in healthy people, an effect of age. I want to bring us back to the point about how much of the disaccharide actually comes through in this type of study. And this was a study done by Bill Fabian and ourselves and Joe Murray a few years ago on children who had environmental enteropathy in African countries and in South American countries, Zambia and Peru in particular. And the main point to show you this is, first of all, of course, there is an increased permeability of the intestine, which is actually documented with recovery of rhamnose. But when you look at the recovery of lactulose, even in these children who have environmental enteropathy and stunting of growth because of the malabsorptive process that occur in environmental enteropathy, even in that situation, only 0.5% approximately of the lactulose was excreted. And in the first hour, which was how this study was done, it was only about 0.1%. Now, I'm not questioning that altered permeability is important in disease states. And I'll just point out some where I'm absolutely convinced that altered permeability is relevant. Shocks and burns, inflammatory bowel disease, HIV infection, graft versus host disease, celiac disease, possibly even pancreatitis. But I think we need to come back and really think, were the studies that were done rigorous enough to be able to conclusively decide that there is truly an effect on intestinal permeability. Here is effects that were seen in experimental animals. I show this slide because it has the names of three of my personal mentors: Vin Chadwick, Alan Hoffman, a distant mentor for me, and Sid Phillips, may he rest in peace. And these were studies that were done in the 1970s using dialpha hydroxy bile acids and showing, using scanning electron microscope, here you see that ursodeoxycholic acid does not damage the epithelium, whereas chenodeoxycholic acid disrupts it in a major way. And this was associated when they gave dialpha hydroxy bile acids like cheno or deoxycholic acid. You can see that's 3-alpha 7-alpha, 3-alpha 12-alpha. You can see the increase in the amounts of the polyethylene glycol 400 moieties that are being absorbed. So this shows that a detergent bile acid can certainly change the mucosal permeability. I also want to draw our attention to a concept and research that was done perhaps originally by Dan Hollander and his groups in the 1980s and 1990s, showing that in patients with Crohn's disease, the family members of patients with Crohn's disease possibly have an increase in mucosal permeability. And this concept was revisited more recently by Jean-Fred Colombel and his colleagues in New York. So this is very much an interesting title. I can almost hear Jean-Fred Colombel mention this. The intestinal barrier, an arbitrator turned provocateur in the IBD. And so he's trying to help us understand that there may be changes in the barrier function that will be important. I'll show you the original data. But apart from these changes in the barrier function with intestinal permeability, there's also evidence, of course, that serum proteins may also be markers in these relatives of patients with Crohn's disease, markers that in the next 5 years, they might be developing Crohn's disease. And you see obvious examples there like ASCA protein, C-reactive protein, amyloid P, and complement C3. So certainly there are serological biomarkers that could be predictors of the development of Crohn's disease in family members. But let's focus on the permeability studies. So in these studies, what they did was they followed patients for up to 10 years, and the most robust data occur up to 5 or 6 years if you look at the number of people involved. And what you also see is that there's a separation in the risk. If you have a lactulose to mannitol ratio, which is normal, you follow the blue line. The risk of Crohn's disease is maybe 2% in those patients. Whereas, I'm sorry, I'm not sure it's at 2%, but yeah, it's going about 2% in those patients. Whereas if the permeability was higher as measured by the lactulose to mannitol ratio, greater than 0.03, then there was a higher propensity to develop Crohn's disease in these relatives. But I want to point out a couple of things. The first point is that the y-axis doesn't go down to 0. So this difference, which looks massive, is really at most a 6% difference. The second point I think I'd like to make with you is that the way the permeability was studied included lactulose and mannitol at doses of 10 grams and 5 grams and 2 grams of mannitol. And we have no idea about baseline contamination or variation between individuals, or indeed within the same individual over time. So I keep an open mind about this, but if you look at the totality of the data that also came from those earlier studies by Dan Hollander and colleagues in the 1980s and 1990s, there certainly seems to be a good hypothesis that there may be a barrier dysfunction predisposition to Crohn's disease in patients in people who are relatives of patients with Crohn's disease. This was another paper recently published, which showed that you can't use serum zonulin to predict that propensity to develop Crohn's disease, at least not using the commercial assay that's available. Third point I want to make, and I'm going to be a little bit faster in the next 3 topics, effects of stress and diet on intestinal permeability. So we know that endurance exercise is one of the factors that has been associated with increased permeability. And this is generally done with intestinal permeability measurements using probe molecules or mucosal damage, as I showed you the markers before. And this is usually biking, marathons, and things like that. NSAIDs, of course, are known to not only cause ulceration, but also to cause an increase in intestinal permeability, measured in this case with urine sucralose to lactulose ratio. Pregnancy itself has been associated with an increase in permeability. I'm showing you the evidence here, certainly in 2 studies, but not in a 3rd study. You all know about the effects of NSAIDs. We know that there's going to be mucosal damage, erosive changes in the intestine, which you see on either a capsule or balloon endoscopic. And you know that several studies, particularly Ingvar Bjarnason and his group at Northwick Park in just outside London in England, had shown in several papers that using either chromium 51 EDTA or saccharide ratios, that there is an increase in permeability with indomethacin, with other NSAIDs, and Cupoletti had shown that lubiprostone may reduce permeability. But this was also very interesting, and that is that if you give zinc concomitantly with the indomethacin, you do not see the increase in permeability that you see when indomethacin is given with placebo. And I'll be coming back to the potential protective role of zinc quite soon. Glutamine is a good nutrient for intestinal enterocytes, and so in the context of conditions like burns, it's been used enterally and it's been effective, whereas if you give it intravenously, it has no effect. In terms of diet and intestinal permeability, Katayun Koshbin and I summarized the literature in this paper, and we came across nutrients that fortify the barrier and nutrients that damage the barrier. The damaging agents, gliadin, ethanol, bile acids themselves, emulsifiers in foods, which serve like bile acids as detergents, and also fructose, which reduces ATP in the enterocyte. On the other hand, several substances, including some amino acids and vitamins, vitamin A, vitamin D, glutamine itself, and fiber through elaboration of, for instance, short-chain fatty acids, can protect the enterocytes and protect the epithelium. I don't have time to go through each one of these, but if you are interested, we have all of this documented in the review in American Journal of Physiology. What about prebiotics, symbiotics, and probiotics? Well, you hear so much about the potential to help the intestinal barrier with these substances. What is the truth? How much evidence is there? And I can summarize all of this by saying that this one paper by Krumbeck and Ali Keshavasian in Chicago is the one that gives the most convincing evidence that a prebiotic galacto-oligosaccharide actually reduces the permeability following aspirin exposure. The other studies really did not show any evidence of prebiotic improvement. And these are the actual data from the Chicago group showing the benefits of prebiotics and also of a probiotic, which we will mention later, which is this bifidobacteria. When you put the two together, you don't actually get an increased protection or synergism between the two. Again, another study that I'm showing in burn patients showing no improvement in gastrointestinal barrier function with a prebiotic. Symbiotics, these are combinations of probiotic plus prebiotic. And again, to cut a long story short, no substantial evidence that these are going to be effective at improving barrier function in response to a stressor, such as physical activity. Probiotics, well, these have several potential modes of action, maintaining homeostasis by enhancing the integrity of the barrier and increasing the production of butyrate, strengthening the tight junction proteins such as occludin and claudin-3. So certainly looks very promising. But actually, when you look at the data, it's really relatively limited in terms of the evidence in critically ill patients in that there's any improvement. In fact, this study showed no difference between viable or sonicated probiotics. Sonicated being therefore just the sort of cell membrane, if you wish, not the active probiotic itself. In aspirin challenge, maybe there is an improvement, as I said, in that study from the Chicago group with Bifidobacterium IVS1. And again, in this study, there really was no dramatic difference, although plantarum, Lactobacillus plantarum may be beneficial. So certainly some evidence of improvement of barrier function. Now, these are studies that have been done with probiotics that didn't actually measure effects on permeability, but we know there is data to suggest that probiotic can help with the healing of ulcerative colitis, maybe in patients with IBD with PSC, or maybe VSL number 3, a multiple organism probiotic combination in refractory pouchitis. What other disorders have been associated with increased permeability? Well, again, these are listed in this paper in Gut from a couple of years ago, and I don't have time to go through this in exquisite detail. But certainly, there is some evidence, for instance, of damage in the intestinal epithelium with increased zonulin in aging. There are also changes in the microbiota, which may also be very relevant. Food allergy, increased permeability. Eosinophilic esophagitis, and I know our colleagues here at Mayo are very interested in this. Increased small bowel intestinal permeability has been also observed in these patients. Non-alcoholic fatty liver disease associated with increased lipopolysaccharide in the circulation, increased lactulose to mannitol ratio, or chromium 51 EDTA. So, and there's a systematic review of meta-analysis that reviews that. This is sometimes associated with small intestinal bacterial overgrowth. Sclerosing cholangitis shows normal lactulose to mannitol ratio, but perhaps more evidence of damage to the intestinal epithelial lining, as shown by the increase in intestinal fatty acid binding protein. TPN, especially if you have enteral deprivation, in other words, patients are being supplemented only with total parenteral nutrition, that is associated, if you take biopsies and you look at the mucosal permeability in vitro, or you look at it under the microscope, certainly evidence of intestinal barrier changes in those patients. Neuropsychiatric diseases have also been associated. Parkinson's disease, amyotrophic lateral sclerosis, and psychiatric diseases. We actually were asked by the reviewers to add some information in our recent paper in gastroenterology. So we put in some pilot data comparing patients with irritable bowel syndrome from those who have elevated 7-alpha-C4, which is a marker for increased synthesis of bile acids. And although this was only based on about 24 patients, we're now currently analyzing the data from 200 patients, of whom 40 or 45 have evidence of bile acid malabsorption or increased bile acid synthesis. And what we see here is there's a trend to those who have increased bile acid synthesis having perhaps more excretion of 13-carbon mannitol compared to the IBS population in general. And as I say, we're hoping to really answer this question with a cohort of about 200 patients. I want to come back to the idea, and that's really quite prevalent in certainly in the if you search this in the public domain, something like Google search, about the role of leaky gut. And this is actually an article by Alessio Fasano from Harvard and GH, and he actually has documented which are the conditions or the disease processes that are associated in human studies in particular, as you can see here, with evidence of a leaky gut. So I'll just mention a few, obviously celiac disease, but also aging, ankylosing spondylitis, diabetes was somewhere. Thought I saw that, yeah, here, type 1, type 2 diabetes, major depression, etc., multiple sclerosis. So it appears that leaky gut is an extremely common problem. And he has a very provocative title to the article, which is that all disease begins in the leaky gut. And so the whole idea is, are there ways in which changes in the local mucosal immune homeostasis occurs because of the leakiness of the gut and the introduction of dietary antigen and other factors that stimulate mechanisms that ultimately result in these diverse disease processes. You also find, if you Google this, that there's a lot being promulgated out there regarding leaky gut and microbial balance. So this is literally quoting something that I found a couple of years ago. When the microbial balance in your gut is right, your whole body functions the way it's supposed to. But when that balance gets out of whack, say because of chronic stress, chronic constipation, exposure to environmental toxins like pesticides, eating a poor diet, or taking an antibiotic that wipes out a lot of those microbes, the bad bacteria cut holes in the fence, and some of them, along with food particles and toxins, leak into the bloodstream. And when your immune system sees organisms where they don't belong, it attacks, causing irritation and inflammation. So they conclude that leaky gut may have many possible causes and cause many possible symptoms. And so you need to remove foods that create the problems, like gluten, sugar, and dairy. And if you're practicing in the motility clinic or the general GI clinic at Mayo, I'm sure you've seen patients who have cut out so many of these dietary factors, and perhaps other clinics as well. Replace the foods with foods that are less likely to irritate the gut, like fermented foods, as shown here. They can heal the mucosa. Repair the damage with supplements, L-glutamine, and I did show you some evidence for that. Vitamin D, there is some evidence for that. Zinc, I showed you the example of indomethacin, and omega-3 fatty acids such as fish oil. Repopulate your good gut, your gut with good gut bacteria. So this is where it's proposed that probiotics may help you do that. And I've shown you evidence that, at least from the literature that I was able to review, that still is not totally clear. Or get a transplant from another person. One of the biggest leaky gut red flags, issues with a variety of foods. And from our clinical practice, I see so many patients who have issues with a variety of foods, but it's not clear that this is necessarily secondary to the leaky gut, and there may be many other reasons for it. Talk with your healthcare provider. You may have leaky gut syndrome. Do not, however, try to treat it yourself, but of course, the next few pictures will show all of the things that you can do with people advertising for substances that, quite honestly, have never been approved for the treatment of leaky gut. But I want to come back on a more serious note to something which I think really does have important promise for the future. And I just want to remind you about the pore pathway through which small ions and small molecules, including probably mannitol and rhamnose, go through. And then the leak pathway, where the mechanisms prevent the passage of molecules normally. But if it's damaged, then you can have molecules of up to 100 angstrom coming through. And this is where, the leak pathway may be more relevant. Of course, if you have damage to the mucosa, apoptosis or ulcers, then of course you have an unrestricted pathway and easy passage of antigens and other injurious materials through the epithelium. But let me focus on a couple of papers which I found really quite interesting. And they come really from the groups of Jerry Turner in particular. And this is an example of an acute tumor necrosis factor-induced barrier loss. And the important thing is that it attacks that myosin light chain kinase dependent path, which is the leaky, not the pore pathway, but the leak pathway. So basically, when treated with either tumor necrosis factor or anti-CD3, which is like an immune model of colitis, you see that the occludin is going up towards the surface of the epithelial cells, indicating the damage to the barrier in that leak pathway. And that's associated with increased leakiness of albumin from the circulation out into the lumen. So if you give an anti-CD3 treatment, the immune colitis, if you wish, in the wild type, you can get all of this albumin. Now, I won't go into this in great detail, but if you then downregulate myosin light chain kinase or caveolin, you actually reduce the amount of leaking that's occurring. But I digress. The important point is that the presence of these markers or of these models of colitis, anti-CD3 or TNF, are associated with increased leakiness of albumin into the lumen. And what I really find interesting is that they've gone on to really dissect this out and find ways to try to restore that myosin light chain kinase. In the interest of time, I'm going to describe this very, very quickly. So if you give TNF, you can get increased albumin flux. I showed you that on the previous slide. But if you give a substance which targets the isomer myosin light chain kinase 1 isomer, that actually prevents the damage to the actomyosin ring, which is important for that leak pathway. And basically what I'm showing you here is that this substance called divertin, which has this property of restoring the myosin light chain kinase, actually reduces the amount of albumin loss, reduces the amount of phosphorylation of the myosin light chain kinase. And when you do a T-cell transfer experiment in inducing colitis, if you don't transfer, this is how the weight of the animal, of the mouse, increases. That's your control. If you give the T-cell transfer with saline, they lose weight dramatically. If you give anti-TNF, they lose less weight, but ultimately they do lose quite a lot of weight. Whereas if you give divertin, it sort of stabilizes the weight loss. So I think this is another interesting example of the application of basic science to potentially change barrier function in the context of inflammatory bowel disease. So in summary, what I've tried to tell you is I want to move on from where Jerry Turner was informing us in 2013 that altered permeability may be an epiphenomenon, yes, and we need to be rigorous about the studies that we do to characterize the altered permeability in disease. Experimental animal models have shown impaired barrier function do not in isolation lead to the emergence of a disease phenotype. In other words, there might need to be more than just the alteration of the barrier. An immune process, for instance, would be necessary. Increased permeability is not necessarily deleterious, but damage to that leak pathway or inducing apoptosis in epithelial cells could be, because you get much larger dietary antigens and other bacterial antigens coming across. So tight regulation is an important goal in human disease. And I think we've moved on in the last 5 to 7 years. I believe we now have a valid measurement for intestinal permeability in vivo in humans. And I think divertin, and there are a few other molecules that are in development that have the potential to reverse the inflammatory process, possibly other diseases, by restoring normal barrier function. But of course, as always, we need large studies and well-phenotyped disorders to investigate. So thank you again for your attention. We do have a couple of minutes for questions if there are any burning questions that you may have. Thank you again.
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.